Hello guys, welcome back to another video. Today our topic is about Proteomics protein identification mass spectrometry. Mass spectrometry or wave. Mass spectrometry is a technique on which gas phase molecules are ionized and their mass to charge ratio is measured by observing acceleration difference of ions where an electric field is applied. Lighter ions will accelerate faster and be detected first. If the mass is measured with previous, then the composition of the molecule can be identified. In the case of proteins, the sequence can be identified. Most samples submitted to mass spectrometry are the mixture of compounds. A spectrum is acquired to give the mass to charge ratio of all compounds in the sample. Mass spectrometry is also known as mass spec or MS for short. Mass spectrometry throws light on molecular mechanisms within cellular system. It is used for identifying proteins, functional interaction, and it further allows for determination of subunits. Other molecules in cells, such as lipid components, can also be defined. A mass spectrometer is composed of several different parts. A source that ionizes the sample, the analyzer that separates the ions based on mass to charge ratio, a detector that sees the ions, and a data system to process and analyze the result. You can also measure relative abundance of an ion using mass spectrometry. Different compounds have differential ionization capabilities, and therefore a density of your ion is not a direct correlation to concentration. Mass spectrometry can be a high through output analytical method due to the ability for mass spectrum to be measured rapidly and with minimum sample handling as compared to GAL methods. It is an analytical method which has a variety of uses outside of proteomics, such as isotope and dating, track gas analysis, atomic location mapping, pollutants detection, and space exploration. History of mass spectrometry. The history of this technique finds its roots in the first two studies of gas excitation in a charged environment. More than 100 years ago, this pioneering work led to the identification of two isotopes of neon, neon 20 and neon 22, Y mass to charge duration decrementation by J. J. Thomason in 1913. Over the next 50 years, the fundamental basic of the technique was further developed. After the coupling of gas chromatography to mass spectroscopy in 1959 by researchers at Dow Chemical, the full potential of the techniques as a highly accurate quantitative method for exploring compounds was realized, spurring a web of developments which continue to the present day. The precision of mass spectrometry led to the discovery of isotopes.